Good day to you all. My name is Guillermo Zavala. I'm with Avian Health International, and I would like to thank Ceva Animal Health for their kind invitation to be with you today to talk about avian metapneumovirus infection. And this will be done from the perspective of the international experience. And you have to know that some of the concepts that you will hear today are based on science, but many of them are also based on practical application in the field. So let's summarize briefly what avian metapneumovirus infection is. And in general, it is a highly contagious disease of the respiratory tract, and it's caused by a virus. It has a worldwide distribution. The etiological agent belongs to the Mononegavirales. That means that it's a single-stranded RNA of negative sense, belonging to the Pneumoviridae family, Pneumovirinae subfamily, metapneumovirus, species avian metapneumovirus. It is an envelope virus, and it is very highly susceptible to mutations. The virus will cause sinusitis, rhinotracheitis, swollen heads, coughing, otitis, and mortality. And therefore, some of the synonyms that have been coined for this particular disease or infection include, not exclusively, swollen head syndrome and turkey rhinotracheitis. The disease is associated with decreased feed and water consumption. You might observe egg production drops, although not very drastic in commercial layers, but they can be fairly substantial in broiler breeders and turkeys. And sometimes eggshell quality can be affected as per field experience. The virus will cause significant economic losses, which can be preventable by live and kill vaccines. And most vaccines contain subtypes A and or B. There are a number of natural reservoirs. This is not a disease where the natural reservoir is the chicken or the turkey. There are many wild bird species that are known to be positive or have been known to be positive to avian metapneumovirus, including ducks, guinea fowl, ostriches, gulls, and a number of other species. Sometimes the documentation has been done by virus isolation, sometimes it has been done serologically, sometimes by RT-PCR, and by any other methods. Now, again, the distribution of the disease is pretty much worldwide, although there are some countries that are considered avian metapneumovirus free, at least in commercial poultry. It is important to know that avian metapneumovirus shares homology from the genetic point of view with human metapneumovirus as much as 80% identity. Turkeys are highly susceptible to avian metapneumovirus, but they're also susceptible to human metapneumovirus. So those concepts are important to know and keep in mind, because when we talk about prevention, epidemiology, disease control, we have to know that not only chickens and turkeys are susceptible to the disease, and unfortunately these viruses circulate in multiple avian species. So as far as the etiology, again, the virus belongs to the Mononegavirales, family Pneumoviridae, subfamily Pneumovirinae, genus Metapneumovirus, and species or type avian metapneumovirus. There have been 4 different subtypes identified in different regions of the world. The most common ones are subtypes A and B, which have been documented at least in Europe, Asia, Africa, and the Americas. Subtypes A, B, and C are now known to circulate, or that have circulated in the United States in different birds, including broiler breeders, broilers, and turkeys. And then finally, subtype D, which has been documented in the European Union, particularly in France, and it has been suspected in Africa and in some Asian countries. So let's look at the virus structure and genome. The virus physical structure and also the genetic material are as follows. Some of the most relevant things that to keep in mind, we will not describe all of them, but some of the most relevant items are the outer proteins. Glycoproteins are the ones responsible for the different subtyping of the virus, A, B, C, and D. There's another important component of these viruses, which is obviously the genome, which again is a single-stranded RNA, negative sense, which we can detect by RT-PCR. Now, from the diagnostics point of view, we will talk briefly later on about what elements of the virus can be used for virus detection or for antibodies or for antigen detection. But the most important elements to keep in mind include the matrix gene, which can be used as a target for RT-PCR. Now keep in mind that because there's high homology between avian metapneumoviruses and other pneumoviruses, perhaps the matrix gene can be practical, but sometimes it can be a little bit difficult to interpret the results. Now, there's another type of gene that is important for us to keep in mind, which is the glycoprotein, which can assist us in the identification of the different subtypes. And then the large polymerase. Now, there are commercial kits available, even in the United States, where the nucleocapsid is the target for RT-PCR. This is currently used at diagnostic laboratories in the United States, but maybe there are some other RT-PCR systems that are being used. So if you can remember that the genome is negative sense, therefore we start reading from the right-hand side to the left, from the 5' end to the 3' end. We have the long or large polymerase. Then we have the glycoprotein, which is very important for subtyping. Then we have the fusion protein gene, the matrix gene, the phosphoprotein, and finally the nucleocapsid gene, which is what a lot of diagnostic laboratories use as a target for RT-PCR identification of the virus. So those are the primary elements that you have to remember. The other thing that we need to remember is that it is an envelope virus. Therefore, the glycoproteins are very important, along with the rest of the envelope constituents. But the practical application of knowing that this is an envelope virus, it means that it is susceptible to the vast majority of disinfectants that we commonly use in the field. So the virus is susceptible to chemical disinfectants, and therefore we should be able to clean, disinfect, and eliminate the virus from the environment. Now, of course, the environment can be recontaminated by different avenues. This is how the virus is distributed for the most part. Probably this is not as comprehensive as it should be, but you have an idea by looking at this chart where the United States is known to be a place where subtypes A, B, and C have circulated. Mexico has documented A and B, Central and South America A and B, Brazil A and B. Europe includes A, B, C, and D, Africa A, B, and D, East Asia A, B, C, and D, and Central Asia A, B, and D. So you can see that very much, for the most part, the virus is widely distributed, and most of the subtypes represented, with the exception of D, are represented in most parts of the world. How is the virus transmitted? The virus is thought to be transmitted via the air, airborne, and by close contact. And the secretions are considered to be highly infectious. The virus survives days in organic material. We have to know that, for example, type C is freeze resistant, and that is important because in the United States and the turkey producing areas, obviously some of these areas are very cold in the wintertime, and therefore we should expect that avian metapneumovirus type C might see a prolonged survival in the environment when the temperatures are very cold. Avian metapneumovirus has been detected in the reproductive tract of turkeys, although the virus is not known to be transmitted vertically. So that's important to know. Shedding occurs until before or shortly after clinical signs. So a lot of the work that was done in the 1980s and 90s very much documented that there was a very short window of opportunity for being able to detect the virus in the tissues of the respiratory tract. And this short window is basically consisting of not more than 3 to 4 days, 3 to 5 days after the onset of clinical signs, after the infection, after the moment of infection. Because a lot of times you can have a significant infection with the virus, but without necessarily observing severe or significant clinical signs. The clinical signs actually come as a result of the consequence, the bacterial infection that is that occurs shortly after the initial virus infection. So many times we lose the opportunity to detect avian metapneumovirus because the virus seems to finish its replication cycle right at the time when we start noticing the first clinical signs. So that's what the research documented in the 80s and 90s and early 2000s. But today we know from practical experience that sometimes the virus can linger for a little bit longer than 5 to 7 or even after 10 days post-infection. How do we know that? We know that by using molecular detection methods, which we didn't really use in the past. Most of the evaluations were done using virus isolation, and therefore, when you use virus isolation, yes, the window of opportunity is very short, but when you use other methods, including molecular detection methods, your opportunity might be a little bit longer than the traditional 3 to 5 days. Shedding, therefore, may linger for days or a week, particularly with subtype B. This is, to my knowledge, not documented in the literature, but when you compare infections caused by type A versus type B, you seem to have a longer opportunity with type B using molecular detection methods. Now, the other thing that's important to know is that reinfection after an outbreak has been known to occur. It doesn't happen very shortly after the first infection. So here we're talking about long-lived birds such as commercial layers and broiler breeders. Here's some of the work that was done in the 1990s, again, based on virus isolation, and in this case, by antigen detection by direct fluorescent antibody test or immunocytochemistry. So you can see that the virus cycle is very, very short, and by virus isolation, we only have maximum until about 5 days before we practically lose the opportunity to detect the infection. This is in controlled studies. Now, of course, in the field, not every bird, not every house get infected at the same time, and therefore we have to consider that as well. Now, as far as antigen detection, you can detect antigen a little bit longer than you can detect the virus by virus isolation. And again, if we were to produce a third graph in this particular study, which was not done, where we would look at molecular detection of the virus genome or genes, then we would see that probably we could detect the virus maybe for about 4 weeks and sometimes a little bit longer after the initial infection. So some of the virus subtype clinical features that have been observed either scientifically or circumstantially in the field. Chicken strains appear to be more pathogenic for chickens. Turkey strains appear to be more pathogenic for turkeys, but chickens and turkeys are both susceptible to both either chicken or turkey strains. Subtypes A and B are cross-protective, and we have seen that multiple times. You can use vaccines against A and protect against both A and B, and you can use vaccines against B and protect against both A and B. What does not happen is that we cannot use subtypes A or B to protect against C effectively, or we cannot use vaccines based on subtype C to protect effectively against A or B or both. So that's an important thing to remember. Now, both types and B can cause rhinitis, tracheitis, pneumonia, otitis, and meningitis. I already mentioned this, but it is important to remember that both types, if not all types, can cause these types of clinical signs or lesions that will result eventually in bacterial secondary infections. Where does the virus go? Avian metapneumovirus is essentially a viral infection of the upper respiratory tract. However, we should expect some air saculitis along with bacterial air saculitis as a consequence of the infection. The infection is not necessarily going to go systemic, although we know that in turkeys, the virus can invade the reproductive tract, and in broiler breeders and commercial layers, there is a clinical component where you might see some drops in production. Now, whether this is the effect, this is the result of direct infection of the reproductive tract, or the result of fever, or drops in feed and water consumption, that has to be determined more accurately. But the fact of the matter is that the virus tropism is essentially for the upper tissues of the respiratory tract, and some of what we see clinically is head swelling. And the head swelling can involve edema and fibrinohydrophilic infiltration, as you see up here in the upper right. These are a number of broilers where there is cellulitis associated with fibrinohydrophilic infiltration after the invasion of bacteria. So the other thing that has to be mentioned is that, yes, particularly in turkeys, the virus has been known to have some tropism for the reproductive tract. So after we have gone through the acute phase of infection with avian metapneumovirus, it's almost unavoidable to see some level of bacterial infections as a secondary or opportunistic infection. And these bacterial infections can be caused most commonly by E. coli, but there's a number of other bacteria that can be involved, and these can be primary pathogens, secondary pathogens. But the thing to remember here is that the more of a disadvantage we have in the quality of the environment, the higher the chances for us to see severe consequences with bacterial infection. And what I mean by that is, if you don't have proper ventilation, if you have significant ammonia levels, if you have dust in the environment, you are more likely to have these consequences. Now, there are many circumstances where you might have excellent environmental conditions and you still see bacterial infection. So one is not exactly 100% dependent on the other, but in general, the tendency is the lower the quality of the environment, the more ability for secondary bacteria to take over. The other thing that's important to remember is that, obviously, as in many other diseases, immunocompetence is absolutely critical. If we have a little bit of a challenge, a little bit of a disadvantage in terms of immunocompetence associated with chicken infectious anemia, Marek's disease, mycotoxins, infectious bursal disease, etc., we can incur into significant secondary infections. So what does the disease look like in broilers? Well, there's going to be a drop in water and feed consumptions. Like many other infectious diseases, the very first observation is there's a drop in water consumption. The second one is, if you can measure it, there's a drop in feed consumption. The birds are taking themselves off of feed. And then we start seeing respiratory signs, and then we start seeing swollen heads, and then we start seeing septicemia and mortality. We're talking about broilers. And when you perform necropsies, you are going to find not a very severe tracheal involvement, not very severe lung involvement. You can see air saculitis like you see, for example, in infectious bronchitis, and you certainly will see swollen heads with cellulitis in that subcutaneous tissue. These are some of the pictures. I apologize for the lack of resolution on the pictures on the left, but that's probably one of the best examples that I could come up with. And then on the right-hand side, you see the classical swollen head pictures. Now, can there be swollen heads caused by other reasons? Yes. For example, in some cases of infectious bronchitis, you sometimes see an occasional bird with swollen heads. But in the case of avian metapneumovirus, it's a lot more of a common observation. Now, let's jump from broilers to grandparents, and that could be very analogous to what you might see in broiler breeders. There's a sudden drop in production, what you see here in your red line. There is almost complete recovery, and then it's like you never saw any case of clinical infection with pneumovirus. But you can see that there's a modest drop in production. In this case, these birds were not vaccinated, and the virus involved was subtype A. Now, obviously, this is not representative of every single case, but it's just one example of the type of drop in production that you could see. It's not as severe as you will see with Newcastle or avian influenza or egg drop syndrome 76, or even a case of infectious coryza in completely naive birds. So this is how the grandparents looked: swollen heads. The birds were not necessarily depressed, they were alert. There was, again, decreased water and feed consumption, reduced egg production. There were some egg shape and eggshell abnormalities. I don't know that those are representative of every single case, but I'll show you the type of abnormalities that were observed in this particular case. And how was this case diagnosed? It was basically diagnosed by acute and convalescent serology. That was very, very clear. There are bacterial complications with low mortality. There's very strong seroconversion, and then the type of virus involved was subtype A. Now here you see a very unusual malformity of these eggs. They look elongated. There's some lack of pigmentation of the eggshell. This particular line tends to pigment the eggshells a little bit more. But the most salient observation is that particular elongation of the eggs. Is that representative of every case? I don't know. I don't necessarily see that very often, but something to consider. Now, it is very different from what you would see with, for example, virulent Newcastle disease. The eggs that you see here on the left, those are obviously from brown layers. In the center, you can see probably a couple of relatively normal eggshells and egg pigmentation. The rest of the eggs are very much discolored. That is very different from what you see in avian metapneumovirus. It's important to contrast this, particularly in countries where virulent or VNDV tends to circulate quite often. On the right-hand side, you see white eggs from single-comb white leghorn chickens, commercial layers, where the eggs acquire that very typical mango shape that is very often associated with genotype 5 Newcastle disease virus, which circulates in North America, or genotype 7, which circulates in Colombia, Venezuela, and Southeast Asia and China, or genotype 13, which is very typical in Central Asia in countries like Bangladesh, Pakistan, India, and a few others. So what is the impact of avian metapneumovirus in commercial layers? And that is a question that many of us constantly have. I myself have a hard time documenting a significant impact in commercial layers. Many layer operations of multiple ages appear to be infected constantly. Birds seroconvert very strongly, particularly after transfer, after onset of production, without necessarily manifesting any significant drop in production, mortality, or lesions associated with avian metapneumovirus. But we have to speculate that many, and probably many of these birds are exposed during the rearing period without much consequence, and by the time they get challenged in the field, they have at least partial protection. So we can't really say that commercial layers are either susceptible or resistant until we do some work to prove or disprove the theory that avian metapneumovirus tends not to be very consequential in commercial layers. But if it does cause an infection, it can cause modest drops in egg production with very low mortality or negligible mortality. There might be a little bit of eggshell discoloration. We're talking about brown egg layers, and for the most part, there's no eggshell, no egg deformities. Now, it is easy for avian metapneumovirus to get complicated with a number of bacteria: MG, infectious coryza, Pasteurella, and a number of other pathogens, as you see here. Now, these pullets, this is a case in Brazil, these pullets seroconverted. They were positive by RT-PCR. You can see that there's severe involvement in the sinuses. But I have to say that this operation also has MG, MS, infectious laryngotracheitis, cholesepticemia, and infectious coryza. So it is really a participant of a respiratory disease complex and not necessarily a primary pathogen in commercial layers, as far as we can tell. Broiler breeders are quite susceptible to infection and to clinical infection. we again are going to see decreased water and feed consumption, respiratory signs, increased secretions, swollen heads, and very characteristically in a relatively low percentage of birds, and sometimes not so low, clinical signs, central nervous system involvement, pretty much as a result of internal otitis and meningoencephalitis. And those can be associated with a significant mortality sometimes in broiler breeders, and that is very typically seen in commercial flocks of broiler breeders that are completely naive, meaning that they have never been vaccinated or exposed in the field at younger ages. And then basically what you would see in a clinical picture is depression, drops in water and feed consumption, eventually birds with clinical signs associated with otitis and encephalitis or meningoencephalitis. Here's pictures from a recent case, very recent case, and the pictures were taken by Dr. Kathleen Seri from the Georgia Poultry Laboratory Network. On the left-hand side, you see an egg pack that is basically normal. Brown broiler breeders don't really pigment very much, particularly depending on the commercial line involved. But if you see all these pictures on the right-hand side, they're all indicative of either meningoencephalitis, encephalitis, or otitis interna, where the birds pretty much acquire this stance of opisthodonus or episthodonus. If I look at these birds, and I am in a country in Central Asia, Southeast Asia, sometimes South America, one of my most important rule-outs is going to be infectious coryza and, of course, Newcastle disease. And much lower in my list is probably going to be low-path avian influenza, either H5N2 or H9N2, or H7N9, which are fairly common in some of these countries. So what do we do for diagnostics? We have several tools that we can use. First of all, our clinical observation, which is going to lead us to suspect avian metapneumovirus infection. We can use serology, particularly acute and convalescent serology, especially in flocks that have never been vaccinated. We can also use tests in the lab to do virus detection. And how do we do virus detection? We basically do virus isolation or detect antigen, meaning viral protein. And then we can do microbiology, not so much for avian metapneumovirus, but to determine what kind of bacterial species are involved in the secondary infection. So as far as the clinical, we already mentioned several times, we are going to be looking for depression, swollen heads, drops in water and feed consumption, perhaps drops in egg production, some respiratory signs, head swelling, cellulitis and subcutaneous tissue, and finally central nervous system signs, particularly in broiler breeders or turkeys. As far as serology, we can use ELISA. We have ELISA kits available in North America that detect subtypes A and B or A, B, and C. So again, acute and convalescent serology using ELISA is fairly useful. But I will have to mention a couple of things about ELISA a little bit later on, because you have to do some interpretation. It's not black and white. And we can use also virus or genome detection, virus isolation in tracheal organ cultures or Vero cells, which nobody does anymore. When we have the ability to do RT-PCR or real-time RT-PCR to detect the virus genome. There are some very few countries that use melting curves. I don't particularly like melting curves because they work very beautifully from the research point of view in the lab and for diagnostics when you have a single entity. But when you have multiple possible agents, it becomes a little bit more difficult to interpret. So RT-PCR is probably the way to go. And in some countries, next generation sequencing has been used as part of the investigations of respiratory disease outbreaks where we don't really have a very clear picture. And one of the multiple agents detected using NGS includes avian metapneumovirus. So, and finally, microbiology, basically to rule out avibacterium, to rule out Pasteurella, and to detect secondary pathogens such as E. coli, ORT, Bordetella, Galibacterium as possible secondary pathogens. So this graph hopefully will help you to kind of picture what is your window of opportunity, what you have to do to diagnose, and at least this is what I have in my mind every time I work with cases in the field, whether I'm in Indonesia or in South America or in South Africa. It doesn't really matter. The general picture is what's going to help us. So we can, number one, detect virus or viral protein or viral genetic material, or we can use the ELISA kits to detect antibodies. So first, we can detect virus. Second, we can detect antibodies. So chronologically, we have to know what is our window of opportunity. At the very beginning, the very first detection that is possible is always going to be nucleic acid, RT-PCR, to detect RNA from AMPV. Second, we can do virus isolation, and remember that we only have a few days. 3 to 5 days perhaps is realistically possible. And then we can detect viral antigen. If you want to complicate your life a little bit and use direct immunofluorescence or immunocytochemistry, you can actually detect viral protein for probably about a week after infection. So first, we can detect virus, and for the most part, we have to consider 5 to 7 days as a very maximum for us to be able to detect virus using any of these methods. Now, I have to mention that in the case of RT-PCR, there is some recent clinical experience where RT-PCR can still detect avian metapneumovirus all the way up to perhaps 2 and even 3 weeks post-infection. Obviously, with CT values that are pretty high, at 14, 21 days, but you have some time, so there's no reason to get discouraged and eliminate the possibility of molecular detection. And then antibody detection. In the United States, we have available, perhaps, I don't know if they're registered or not, but we can use a BioCheck or IDEX, which detect subtypes A, B, and C. And overseas, there are 2 additional ELISA kits that are very commonly used, particularly IDVET out of France, which detects subtypes A and B. And then there's HIPRA out of Spain, which also detects A and B, but that particular ELISA kit is not available commercially. It's just used internally by the vaccine manufacturing company. So we have 4 different options in the international field. So what do we do? How do we sample birds? We are going to collect tracheal and or oropharyngeal swabs from 11 live chickens, 11 dead chickens, and even better, 11 random chickens. And you can make pools of those, and for every 11 chickens, whether they are live or dead or random chickens, place the samples, place the swabs in BHI, brain heart infusion, transport that in refrigeration to the lab, and request virus isolation and or RT-PCR. And remember that if you do want to detect the virus, the nucleocapsid gene is very, very practical. If you want to do sequencing for an estimate of whether you're dealing with A or B, well, then the glycoprotein is gonna be your target. And then for detection of antibodies, we are going to collect serum for acute serology immediately after the first noticing the first clinical signs, and then we can do convalescent serology 2 to 3 weeks after infection. Of course, in broilers, this is not going to be possible because they tend to get infected, probably during the 3rd, 4th, 5th weeks of age, so they might not have enough time to seroconvert, but we have to attempt doing that. So those are essentially the most practical ways to approach diagnostics. So this is just a quick reference. If you need to detect complicating bacterial species causing otitis and meningoencephalitis, a very practical way is to section and separate the cranium from the vertebrae and introduce your swab and go around the cavity generously, and then use that to plate on blood agar, McConkey, whatever you normally use, to try and detect what bacteria is actually causing that meningoencephalitis. And as far as the swabbing, very much like what you do for a number of other respiratory agents, which is oropharyngeal swabs, or in this example, in this picture, a tracheal swab. So let's talk towards the end about prevention and control. So biosecurity is generally ineffective. It's not one of those diseases where if we implement or improve our biosecurity, we're going to succeed in preventing infection. Of course, you have to practice biosecurity to prevent many other potential problems, but we should not expect that biosecurity is going to be a very strong weapon against avian metapneumovirus. Good management practices is probably a lot more reasonable for us to think about. How do we optimize ventilation, sanitation, and poultry husbandry with good management practices? Control of immunosuppressive disease, very important. Of course, no disease is going to be managed properly when we have immunosuppression. Anti-mycoplasma strategies. AMPV is really not compatible with operations that are MG positive, and that's unfortunately something that we see very often in commercial layers. Those are the types of flocks that tend to get in trouble much more easily. Control of other respiratory agents, very important. And then finally, consider vaccination where vaccination is legal and available. So there are live and kill vaccines. Many countries have registered both live and kill vaccines. Some countries have only kill vaccines registered and approved for use. And these vaccines can be used in turkeys, in broilers, layer breeders, commercial layers, and broiler breeders. And of all those, I will mention a couple of things later on, but you have to be really very careful and justify very well whether you want to vaccinate broilers. But broiler breeders, layer breeders, and commercial layers, you can easily justify vaccinating with live and killed, or at least with a kill vaccine. So we're not going to go through the details of the immune responses against metapneumovirus. Let me just mention that a couple of things that are important to remember. Avian metapneumovirus obviously is going to infect birds, and antigen presenting cells, macrophages, dendritic cells, and perhaps even somatic cells, will present processed antigen to helper T cells. And the two types of responses that you can expect from a live infection include Th1 responses, which are going to result or derive into activated cytotoxic T cells to mount a cellular immune response. This tends to happen, as in other diseases, fairly quickly, 3 to 5 days perhaps. But in the case of antibody responses, humoral responses, you can also see a robust Th2 response resulting in the production of IgM, IgG, and IgA. Now, in the case of avian metapneumovirus, IgM is going to be important for protection of systemic infection. Now, I already mentioned that this is not necessarily a systemic infection. IgG is also systemic, but it helps to protect the reproductive tract. And finally, IgA, which probably is one of the most important immunoglobulins that participate in the immune responses, where in the respiratory tract, in the mucosal membranes of the upper tissues of the head, and so the combination of some antibody response and of cytotoxic T cell responses, cellular responses, are going to be together in concert participating in immune responses against live infections. When we're talking about kill vaccines, we essentially stimulate more than anything IgG, not necessarily a significant amount of IgM or IgA, and certainly not secretory IgA, which is not going to go to the mucosal membranes. So very important to consider. Live virus infection in the field triggers humoral and cellular responses. Live vaccinations trigger both humoral and cellular responses. Kill vaccines will trigger essentially antibody responses and mostly associated with IgG. So what vaccines and vaccination do we have available in different countries? Well, we have live attenuated vaccines and inactivated or kill vaccines. And I already mentioned the types of responses that you should expect. Live vaccines include A and B subtypes. Inactivated vaccines also include A and B subtypes, and those also are available in multiple countries. This is a chart that is probably very incomplete. I'm not sure, but it's an example of the multiple different vaccines that are available overseas, outside the United States, with different strains from different sources, whether it's a turkey strain or a chicken strain, and also different subtypes, A or B. And I don't know if there's a vaccine available for C. I assume at some point a vaccine was produced, but overseas, basically it's subtypes A and B. And again, there are chicken strains, turkey strains, or both. So now let's look at a few examples of what you should expect with serology in countries where avian metapneumovirus circulates routinely. This is in Brazil, commercial layers, and there are multiple flocks represented in this graph. This is not a single graph chronologically of a single flock. Every bar represents a different flock, except that they have been sorted out by age. 7 weeks, 13, 20, and 50 weeks of age. You can see that there's gradual seroconversion in the absence of vaccination in an operation that doesn't really have any clinical signs that would make us suspect avian metapneumovirus infection. So you can have circulation of the virus, you can have seroconversion in the absence of clinical disease. This is another operation, also in Brazil, that routinely vaccinates against avian metapneumovirus, that routinely has respiratory problems that sometimes can be attributed to AMPV, but sometimes to other disease agents. You can see that there's also a tendency of seroconversion, increased seroconversion during production. One thing that I forgot to mention and point out is the fact that once the birds seroconvert, whether it's vaccination and or field infection, the seroconversion is quite robust. You can ordinarily see titers about 15,000 all the way to 25,000, very much in this range. This is representing GMTs, but you can see that the maximum titers can reach 45,000, 40,000, 35,000, 25,000. So it's not uncommon for you to see very strong seroconversion. This is another operation. These are broiler breeders. They receive only one kill vaccine. There's no live vaccine approved. And you can see that there's a gradual seroconversion in many of these flocks. There's a few flocks that get challenged early in rearing, and it's pretty obvious because they have titers of 20,000, 20-something thousand. Here's an example of broiler chickens in Central America, Panama to be precise. These are birds of 5 and 6 weeks of age, just a few flocks of broilers. And you can see that the titers are below the cutoff ratio. One of the kids has a cutoff value of about 300, 350. The other one has about 1,200. So that's another thing to consider. Turkey breeders, Chile, one killed vaccine, occasional clinical signs associated with avian metapneumovirus, but not diagnostics are not often done on these birds. But you can see that there's a gradual seroconversion. The birds reach over 10,000 in some cases. This is in South Africa. These birds received one live and one killed. These are broiler breeders. And you can see that the titers are fairly robust, about 17,000, and in this case 21,000. Very much in line with what we see in South America and South Africa, North Africa, Central Asia, Southeast Asia, and perhaps also in the European Union. So how does industry vaccinate when they do vaccinate against avian metapneumovirus? For long-lived chickens, commercial layers, and or broiler breeders, in the case of commercial layers, you can vaccinate with live vaccines at around 4-6 weeks of age, and then at 12 to 14 weeks of age. And pretty much you would vaccinate the way you vaccinate for bronchitis or Newcastle in this case. And then you go back with kill vaccines at around 12 to 18 weeks. What's most commonly done is to give one single kill vaccination. Very few companies actually vaccinate twice at 12 and 18 weeks. So the norm would be 2 live vaccinations, 1 kill vaccination. In the case of turkeys, whether it's tom turkeys or breeders, Live vaccines can be given basically for breeders, and sometimes also they will receive kill vaccines. So there's a number of things that we can face in the field. There's sometimes you vaccinate and you do have significant complications, significant vaccine reactions, bacterial complications after live vaccinations that are done, particularly by spray, and very importantly in areas or farms or houses where the environment was not exactly adequate, or where you might have ancillary problems that have nothing to do with pneumovirus, but that can really predispose or complicate the situation. But it's sometimes difficult to conduct investigations as to what might be not going right when you vaccinate your birds. I would say that for the most part, live vaccinations, kill vaccinations in long-lived birds, broiler breeders, layers, are very much necessary, are very beneficial, are very supportive of your control programs for avian metapneumovirus. And realistically, vaccination is a lot more likely to succeed than trying to improve your biosecurity and things of that nature in every single situation. This is much more realistic to be able to vaccinate and protect your birds. In the case of broilers, my philosophy, and this is only personal, my philosophy is to leave them alone and to work with the environment, with good management practices, with immunocompetence, and as much as you can to avoid vaccinating these birds in the field. However, there is vaccination of birds at the hatchery in combination with bronchitis and or Newcastle. And some of the vaccine manufacturing companies are advocating the mixing of these different viruses, including avian metapneumovirus, whereas other vaccine manufacturing companies are completely opposed to doing that. So in that case, they recommend vaccinating a few days later, 10, 12 days in the field against one or the other. So again, it's a personal choice, and based on experience, I would say that layers and breeders, by all means, it's difficult to have significant complications with either live or kill vaccines. Broilers, it can be a little bit controversial. So with that, I would like to thank you very much for your attention. Hopefully this will help you with your general picture. Remember that this is only kind of a personal perspective on based on experience in the international field, and also based on science related to avian metapneumovirus. Thank you again, and thank you, Ceva, for the invitation to be here today with you. Goodbye.